Welcome to today's session in our webinar series on emerging issues in the coronavirus pandemic. Where I'm sitting right now in Philadelphia, the skies are dark, it's been thundering, and we're under a tornado watch, so I'm hoping that our internet connections are not disrupted. For information about upcoming webinars, as well as archived versions of our previous webinars, please visit drexel.edu/Dornsife. Our webinar today is titled, "Why do we need clinical trials and what will they tell us?" Our previous webinar examined vaccine development, evaluation, and licensure, both domestically and globally. Today, we continue our examination of promising interventions and will focus on possible treatments for COVID-19. Our two panelists, in the order that they will speak, are Dr. Leslie McClure, who is professor and chair of the Department of Epidemiology and Biostatistics at the Dornsife School of Public Health. Dr. McClure is a biostatistician with diverse research interests, including the design and analysis of randomized clinical trials, which is informed by her extensive experience leading multiple clinical trials coordinating centers. Dr. McClure is a fellow of the American Statistical Association and of the American Heart Association, and is on the board of directors for the Society for Clinical Trials. Dr. McClure will provide an overview of the importance of the role of clinical trials in evaluating new medications or new uses of existing medications. Our second panelist is Dr. Charles, or Chuck, Cairns, who is the Dean and Senior Vice President of Medical Affairs at the Drexel University College of Medicine. Dr. Cairns is an internationally recognized leader in emergency medicine and critical care research. He previously served as the principal investigator of the National Collaborative for Biopreparedness, funded by the Department of Homeland Security, and as the director of the U.S. Critical Illness and Injury Trials Group, funded by the National Institutes of Health. Dr. Cairns will provide an update on efforts to identify and develop COVID-19 therapies. Each of our panelists will speak for about 10 minutes, then we'll have time for questions, including questions submitted in advance, as well as questions that you can submit during the session. As moderator, I'll do my best to make sure we get to as many of them as possible before the end of the hour. One caveat is that offering individual or personal medical advice is outside the scope of our charge today. Such questions should be addressed to your healthcare provider. Dr. McClure, I'll turn it over to you.
Thank you, Jim, and thank you to my colleagues at the Dornsife School of Public Health for including me in this webinar. It's my pleasure to speak with you. I will start off by apologizing in advance if there's a dog barking or a child speaking in the background. I've asked them all to be quiet, and the kids sometimes listen better than the dog, but not always. So I'm going to start off with some information that's been in the news lately. Last Friday, the New England Journal of Medicine published a paper by a large collaborative group in the US and around the world examining a drug, remdesivir, as a treatment for COVID-19. In this paper that was published, they concluded that this cohort of patients hospitalized for severe COVID-19 who were treated with compassionate use remdesivir, clinical improvement was seen in 36 of 53 patients, which is 68%. They then go on to say measurement of efficacy will require ongoing randomized placebo-controlled trials of remdesivir therapy. So there's quite a bit of buzz surrounding this paper in social media and in the media in general. But the reality is that the results don't really tell us much. And perhaps the most important conclusion that they make is that there is this need for randomized placebo-controlled trials. Although we don't need this paper to remind us of the importance of clinical trials, they do a good job at least of focusing on that. So I'm going to unpack their statement today, focusing on two aspects of that. randomization, and placebo control. Then I'm going to talk about the length of clinical trials and some approaches we have to shortening them. So let me first start by talking about placebo control and what this means and why it's important. In the remdesivir paper, the authors report that 68% of patients showed an improvement in oxygen support, whereas 15% showed worsening. While 68% sounds impressive, although the authors do attempt to compare their results with those from other studies, we don't really know anything about what would have happened in other contexts. So it's possible that those same 68% of patients would have improved without remdesivir. On the other hand, they indicate that 15% of patients treated with this medication worsened. Again, it's not clear what would have happened to these patients had they not been treated. Ultimately, what our goal is when we do a clinical trial is to determine what one's outcome with treatment would be compared to what their outcome would be without treatment. But we can never observe that. We can never see what happens to someone when they get treated and also when they don't. So because we can't see that in a single person, it's necessary that we have controls. Further, it's not enough to simply have a control group, but also that patients are randomized to receive the treatment or the control. That is, for each patient that enrolls in the trial, they're equally likely to receive the treatment or control. I'm going to share my screen with you now to show a graphic that might help understand randomization a little better. Okay, so suppose we're doing a trial where we have two treatments or a treatment and control as shown on the screen. We have this patient population comprised of patients that are red stars and blue squares. Suppose that all of the red stars took the treatment and all of the blue squares got the control. If we were to see a difference in treatment and control, we would not know whether that was due to the treatment itself or whether that was due to the fact that all of the red stars got the treatment, all of the blue squares got the control. So perhaps there's something different about the red stars and the blue squares that led to this difference. On the other hand, if we randomize the blue squares and the red stars to each of the treatments and have some sort of balance between them, then we can then say that the difference might be due to treatment, should we observe a difference. So it's important that we have a control group, and it's important that we randomize to our control group. But it's not always easy, and it's not always fast. If you listened to the webinar last week on COVID vaccines, Dr. Wentworth spoke about the timeline for clinical trials for vaccines. She set us up really nicely for our webinar today. Just as with vaccines, approval of new drugs for treatments take a long time. So I'm going to show you another figure that hopefully our pictures are not covering up that shows the timeline. This is from a paper in 2011 in the British Journal of Pharmacology that shows the timeline of the drug discovery process. Now this is from the very beginning. So at the very beginning, there's basic research to pick chemical compounds that might one day become a drug. And this takes years and years. And then there's a lot of work that needs to be done before this compound ever makes it to humans. Oh. So if you look at what's in the blue box, all of that happens in animals and in the lab before a compound ever makes it to humans. In the red box is the part of the drug discovery process that occurs in humans. The average time in total is about 12 years from compound discovery to approval, and about 1 in 1,000 compounds that are explored ever make it to human testing. Once a drug enters human testing, this red box here, it's about 6 years to FDA submission. So those are that includes all of the time it takes to test a new drug in people, and then all of the bureaucratic process that it takes to get approval from the FDA, which often can be 2 years or more. Only about 10% of drugs that enter human testing, that enter this red box, eventually get approved based on a study from 2011. So this process is long and arduous. And one reference I found indicated that the filing for the FDA, the paperwork that this requires, can often be over 100,000 pages. So what do we do when we're in a situation like this, where we have some urgency to develop therapies and treatments for diseases like COVID-19. How can we speed this process up? So one approach is through statistical innovation, such as adaptive designs, which allow us to do things such as look at multiple treatments in a single study or multiple doses of a single treatment to be explored in a single trial rather than looking at each separately. So in an adaptive design, we can go through time, we can eliminate treatments that don't show promise early on or doses that don't show promise early on, and which allows us to focus on those that do. These type of trials help, but much of the slowdown is surrounding other aspects of the process of drug development. So there was a nice op-ed in last week's Washington Post by a colleague of mine at the University of Florida, Natalie Dean, about other ways that we can speed up the process of getting new treatments to patients in times like this. It's really, really an exciting time to be somebody who does clinical trials, and particularly someone who does clinical trials in infectious diseases. But the information is changing daily. And so keeping up with this can often be challenging. One way to speed up the process is to assess drugs that are approved for other uses. So this is mostly what is happening right now with the drugs you're hearing about in the news. This allows drugs with known safety characteristics to start later in that process I showed, but still requires approval for new uses. So one of the aspects of the trial processes that take a long time is the development of the protocol, which is the roadmap that describes who will be included in the trial, what data will be collected, and how will that data be collected. To speed this up, one approach is to borrow from protocols for trials in patients with similar diseases, such as pneumonia. Additionally, getting approval to do research in humans can take time, and often each location at which the research will happen will have to have its own approval. So for instance, at Drexel, we have an institutional review board that approves all human subjects data or research. I'm on it, actually. We meet monthly. And so to get something approved in a faster manner can be difficult. So think about this. If we want to implement the same clinical trial at several hospitals, such as the Remdesivir study did, each one needs approval to do research in humans. This can take considerable amounts of time, lengthening the timeline until a new treatment can get into the hands of doctors. Use of a common institutional review board can help eliminate this issue. And actually, I spoke with the chair of our IRB, Institutional Review Board, this morning, and another way we can help speed this process up is through the use of reliance agreements, and we can expedite those considerably. Once the trial is underway, its success is dependent on the path of the epidemic. So since the spread of the disease will dictate the timing of cases being seen in different hospitals, in order to make the most of all of the data, it's essential that each hospital be ready and able to share their patient data rapidly to help answer important questions more quickly. This isn't as easy as it sounds. Even in the age of electronic medical records, different hospitals use different systems to collect their data and to collect information on their patients. Sometimes abstracting that can be quite challenging, and sharing that when there's sensitive patient information can be quite difficult. Then once the results are ready, and assuming there are encouraging results, it's crucial that approval by the FDA happen in a timely fashion. In the case of coronavirus, the FDA has created what they're calling the Coronavirus Treatment Acceleration Program, CTAP, to expedite the development of COVID-19 therapies and to assess whether they are safe and effective. According to the FDA's website, the CTAP uses every available method to move new treatments to patients as quickly as possible, while at the same time finding out whether they are helpful or harmful. So to be fair, I did a little research trying to really see what CTAP involved, and there wasn't a lot of information on the website. Another mechanism for moving trial or treatments through the clinical trials process more quickly is the COVID Collaboration Platform, a cooperative initiative out of the Johns Hopkins School of Public Health, for which we at the Dornsife School of Public Health are a sponsor. This initiative will incorporate many of the approaches to speeding up trials that I described above, including developing common protocols and sharing them, implementing common human subject review, and common approaches to assessing trial success, which also includes evaluating and defining outcomes in a common fashion. The last thing I want to mention before I turn it over to Dr. Cairns is the reminder that the goal of clinical trials is to find effective treatments, but this must not outweigh the risk posed by the treatment. The president has spoken about the use of hydroxychloroquine, a malaria drug also used to treat other conditions, as a potential treatment for COVID-19. This drug has not been evaluated conclusively in randomized clinical trials for COVID-19 and is currently being used off-label, which means it has been approved for other uses. This is perfectly legal. That is, the FDA allows physicians to prescribe medications for uses other than what they were approved to treat. There's a strong desire by physicians and patients to have some means to treat COVID infections, and rightfully so. So this is one approach they're trying. However, hydroxychloroquine also carries risks that could be either more mild or more severe, and it's important to understand the trade-offs between those risks and the benefits. Rigorously conducted clinical trials can help us determine which way the balance tips. Chuck, I'll turn it over to you now.
Well, thank you very much, Leslie. And first of all, thank you, Jim, for putting this panel together. And thank you to the Dornsife School of Public Health. It's an extraordinary resource, not only for Drexel University, but frankly, for the rest of the country, if not the world, in terms of public health. And so my background is in clinical trials, and clinical trials specific to challenges like pandemics, as well as in critical care and emergency settings. And clearly, one of the aspects of the COVID-19 pandemic is that people become very seriously ill, requiring intensive care unit care. And when that fails, unfortunately, these cause fatalities. And so the remarkable aspects of this pandemic response have been the number of people, the number of groups, the number of investigators, the number of clinicians, the number of public health authorities, and frankly, the number of companies and governments around the world to try to take this on. And in fact, I did a quick count of the number of clinical trials right now for COVID-19, and I am at 304. So there is enormous activity in terms of the number of clinical trials to take on the challenge of this virus. Now, this virus is new. There are no approved treatments for this virus. And I just want to repeat that. There are no FDA approved treatments for this virus. Frankly, there are no proven treatments for this virus. And so we are in an area of uncertainty. Now, there is hope. And that, of course, is what's driving a lot of the press you hear about treatments. And frankly, it's being used every day, including in hospitals in Philadelphia and around the world. And so I'm going to just categorize some of these efforts in terms of what they're intending to do. Dr. McClure gave a wonderful overview of the recent experience with remdesivir. Remdesivir was designed to be an antiviral drug. It was designed against drugs like MERS, like SARS. And as background, COVID-19 has another name called SARS-CoV-2. It is very similar to SARS-CoV-1, the virus that caused the huge outbreak around 2003. It also shares characteristics with MERS that came out of the Middle East and created a number of challenges, very similar in terms of respiratory disease and need for intensive care. So this class of viruses called coronaviruses have been studied in some detail, and treatments have been developed against it. So the obvious first choice, well, what about those treatments? And remdesivir is a classic. If you take the virus and put it in cells and you expose it to remdesivir, it turns out that it inhibits its growth. So the idea of using remdesivir is it showed activity against MERS, it showed activities against SARS, it should show activity against COVID-19, and it does. But those are in vitro studies of isolated cells. There frankly isn't a lot of experience with remdesivir in the coronaviruses that we just mentioned. And so most of the experience is in its use on a compassionate basis, as Dr. McClure talked about. And so that's anecdotal, it's very patient specific. And then even the accumulation of clinical knowledge from those individual patients merely tells you that the drug can be given. It might tell you that it can be relatively safe. and that there may be an opportunity to explore it further. And I think that's why we need to be able to get more experience in this drug across a wide range of patients at a specific dose, a specific time, because frankly, most times that people get compassionate use drugs is when they're pretty late in the illness and you've tried everything else beforehand. So I think that's my first comment about remdesivir. But remdesivir is a good target because of this history and how it's developed. Another way to take a look at how we might treat this virus is to think about what are drugs that work against viruses similar to remdesivir. And we do have a lot of experience with drugs like this. We have drugs that we've used in influenza that have been shown to have some benefit, although I want to emphasize, COVID-19 is not influenza. So just because we have an antiviral drug that works in influenza doesn't mean it will necessarily work against COVID-19. But there certainly are clinical trials of drugs like Tamiflu you may have heard of, and other kinds of novel anti-influenza drugs, some of them out of Japan, that have shown, again, very similar anecdotal evidence in case series that when given to patients, they appear to improve their symptoms versus a control group. The problem with the control group is it's another antiviral medication, which has not been tested in coronaviruses either. So I'm not sure what those data mean. The next kind of example would be, let's not work on drugs that are known to impact viruses like influenza, but are known to work on other viruses that we know can have impact and save lives. You can get into the HIV virus inhibitors. And the big class has been HIV protease inhibitors. In this case, the virus comes in, it actually creates a protein that helps it develop and create its own response. If you can inhibit the development of those proteins through this protease inhibitor, then you theoretically could stop the impact of infection. Those are being tried right now with COVID-19 on a pretty large scale. The challenge is some of those initial trials have not shown a benefit of those drugs. And again, COVID-19 is not HIV. It's not influenza. It is completely novel. So then the thought comes down to, are there ways to inhibit general viral replication? Keep the virus from infecting cells. That's how we get into this chloroquine and hydroxychloroquine issue. You know, that's a very old drug. Chloroquine's been around since the 1930s, and it's been used for malaria, and hydroxychloroquine was used to kind of decrease toxicity, and has been used for lots of different diseases that involve inflammation. And in those studies, when you look at how hydroxychloroquine or chloroquine works, it's not entirely clear, but it does have an impact on a way a virus or a parasite gets involved in a cell. And once it's in that packet, it then is dependent on a certain pH or acidity in order to release, whether it's the parasite or the virus. In this case, hydroxychloroquine and chloroquine create a different pH. It makes it more alkaline. And so that is thought to inhibit then the release of that parasite or virus. We don't know if that for sure that's the case in terms of COVID-19, but there are some studies in isolated cells that suggest that can happen. The challenge with the clinical data on hydroxychloroquine is, again, very similar to the experience that Dr. McClure went through with remdesivir. That's the state of the data. It's been given to a series of patients in both China, from Chinese researchers, from China, for French researchers, and they show that there may be some benefit, but they're not randomized controlled. We frankly aren't sure of the dosing, and I still have concerns about the timing of when this drug is given. Remember, we aren't talking about a controlled trial when we go with a diagnosis, or we go with a particular time frame in hospitalization, or with follow-up. And so I think these are really important questions, because no drug is without side effects, if it's at all effective. And certainly chloroquine and hydroxychloroquine have side effects. In some cases, they can be lethal. And so I do think that's one of the reasons you hear a lot of scientists saying, we need more information. We need to better understand how it would be used, but most importantly, we need some outcomes that we can have confidence in before we extend it to the care of our patients. I think the last series of approaches has to do with the body's immune system itself, even beyond the virus. And there are two very interesting approaches. One approach is to actually look at what the inflammatory cascade is. And that's a long word, just to basically say, is that once you become infected, your body starts a chemical reaction called inflammation. During that period of inflammation, these chemicals go throughout your body and they can do damage to organs. They can do damage to systems like your lung. Of course, they're needed in order to be able to kill off the virus or kill off the bacteria. So it might be possible to allow that inflammation system to kill off the virus or the bacteria while not causing damage to the lung or other functions. That is a new area of research. There are some drugs available. The one that's received the most attention is the one against a cytokine, that's what these chemicals are called IL-6. And then the final one, the one that there's been a lot of press about recently and the FDA announced an investigation of, and that is using convalescent plasma. The idea behind this is that the body's immune response can be captured in people who've recovered from the virus. So the basic concept is the virus comes in, you develop antibodies against the virus as part of your defense system. The person then recovers, but those antibodies remain. Those antibodies can then be harvested through concentrating plasma. And if appropriate safeguards are given, those antibodies can then be given to another person with the infection. So you're basically transferring the immunity from one person and the successful response to another who hasn't yet generated it. It's a very exciting area of work, but you can imagine there are lots of challenges. The first challenge is you need to have a lot of plasma. The second challenge is you got to make it safe. And the third part is that we're not sure which are the exact right antibodies. And I must tell you, there's questions about neutralizing antibodies in this virus. I've been involved in these trials for influenza. Our first experience was wonderful. In fact, I had a patient who turned around within 12 hours of receiving this. He was otherwise on a ventilator and was about to die. The problem is when we did the large phase 3 study of more patients, we found no benefit. So clearly we need to learn from this experience as we move to this next phase of COVID-19. So thank you so much, Leslie, for the opportunity to be here. Look forward to any questions that you might have.
And thank you so much to the both of you. I mentioned at the start of the seminar that it was cloudy and now the sun's shining in my neighborhood. So you guys must have done a great job. Thank you. We've received a number of questions. The first concerns the trade-off that you mentioned, both of you, between the amount of time it traditionally, historically takes to move a drug through all the stages to approval and the urgency here. This was a theme also in last week's conversation about vaccines. So the question concerns how can we be sure that these expedited procedures are going to be giving us good information, both about the efficacy and safety of new drugs or vaccines?
Well, I can start with an answer to that one. I'd say this is a very important question because we want to make sure that not only do we respond to the crisis, but that all the drugs interventions we develop are both safe and effective. I think that the process for determining whether a drug is safe or effective have been well delineated by Dr. McClure. And I think that those steps are necessary, but we can speed up those steps. We can accelerate some developments. There are ways that we can do it through new clinical trials designs. As she mentioned, this adaptive trial where you actually change the parameter of the trial as you learn information from it. You learn who is best suited to receive that treatment, frankly, who gets toxicity from that treatment. You can identify that early on and start to narrow down the population that would benefit. You can also start to get a better idea of effect. And you might be able to stop that trial significantly earlier because you figured out who benefits, who doesn't, and what the basis of the effect is in terms of magnitude. But it will be critical for any of these processes to still take a look at safety, to still take a look at efficacy or the ability of the treatment to have a positive outcome. And no matter what happens, am sure that those steps can be done. They're absolutely necessary, but they can be sped up. And clearly the FDA has been a world leader in this. And the FDA clearly is paying attention to all of these treatments, at least that are released in the United States. I hope that answers the question.
Yeah,
I think so.
Yeah, I would just add that we don't want to mistake speed for lack of rigor. That just because the processes are moving more quickly doesn't mean that the same attention to detail isn't being taken in reviewing drugs, in collecting data, and in reporting. And so I think, there is a lot, and part of this includes peer review, that process of people submitting their research for others to review. And I think that process is still in place. The FDA is still using their top-notch scientists to review these data. They're just doing it at a different pace. So I wouldn't mistake speed for rigor.
Great, thank you very much. Our next question concerns observation that people of minority race ethnicity, particularly African Americans and Latinos, have higher rates of severe illness and higher risk of death if they acquire COVID-19 infection. So the question concerns, how can we be sure that the clinical trials enroll people from different racial or ethnic backgrounds to make sure that we're evaluating the efficacy and safety of these drugs in different groups of people? And as part of that question, just say a little bit about what's the process of assuring diverse representation in clinical trials like these?
So I can start with this. From the NIH perspective, it is part of the process when you have a trial that you propose, that you explicitly describe how you're going to increase the enrollment among underrepresented minorities or among people who are disproportionately affected by a disease or condition. So that is explicitly part of the funding requirements in order to run an NIH trial. Now that said, this is something that for sure is an issue and a concern. And actually, I had made a note before the question came up to mention that, as several of my colleagues are leading the way, particularly in Philadelphia, in looking at the rates among different race and ethnic groups. And so it's, of course, particularly a concern for us at Drexel, who are concerned about health disparities. So I think the other part of that, and Chuck can probably speak a little more to this, is that at Drexel, we're involved in an initiative to increase enrollment in clinical trials of people from a variety of race and ethnic groups. And I think the probably broader answer is that it's contingent on investigators to work hard to explain the process of the trial to all potential participants and be inclusive in their work.
Thank you. Chuck, you want to add to that at all?
Yeah, I'd like to, Jim, because it's a really important issue. And I think there are multiple layers of importance here. I'd say the first one is that we need to better understand this virus. We need to better understand what happens with infections. We have people who clearly get the virus and have minimal or no symptoms. We have people who get the virus and have a mild disease and get better. And then we have people who end up having these complications, requiring an intensive care unit and even dying. And we don't really understand that variability very well. Frankly, we don't understand this age disparity. It's clear that older people are dying at a higher rate than younger people, but clearly some younger people have the same kind of need for intensive care treatment. And then finally, we need to better understand each individual's response based on their genetic background, based on prior exposures. And frankly, we're part of a group from the National Institutes of Health to take a look at this immune response. It's the NIAID immunophenotyping study, and it's just a fancy way to talk about how do we characterize how this virus impacts the immune system in individuals. So clearly, we need to better understand this virus. But clearly, when you get a signal like this about racial disparities, we need to address that as well. One of the issues beyond what Dr. McClure said is that I'm involved in research that's in emergency departments and intensive care units in hospitals. And so we just try to enroll everyone who's eligible in these trials. And one of the things about emergency departments and critical care is that people who require specialized services usually do get concentrated in hospitals and intensive care units. And so at least we'll have an opportunity to get a pretty wide group. But that's a very select group who are getting the virus. These are people who are severely impacted, hospitalized, and require intensive care. But we need to better understand this virus, its impact on populations. And as Dr. McClure said, we're well positioned here at Drexel. Our partners include St. Christopher's Hospital for Children in North Philadelphia, our partners at Tower Health in suburban areas, and our partners with Tower Health in Reading in a rural area, all of which have suffered dramatic challenges to life expectancy and morbidity and other conditions. And we intend to have our systems enroll all these people. And we want to be very active in the community in providing that kind of opportunity for all people across all communities so we can benefit the most in this country and around the world.
Thank you very much. It's clear there's a strong commitment to that important question. One of the questions that we got before the seminar started concerned observations in South Korea that were recently published that individuals who'd been infected with COVID-19 tested negative, including 2 successive tests separated at least 24 hours apart, who then later tested positive again. So the question is if this phenomenon is verified through further observation to be something that is really happening, how would that affect the definition of endpoints in clinical trials?
Well, I could take it on from the pathophysiology side. I'm interested in Dr. McClure's answer to this. As I said, I think we need to get a better understanding of this virus and the body's defense system. Frankly, there have also been reports that people who've been infected, who get better, don't have any detectable antibodies. So is there something about this virus that's impacting the immune system in a different way? There certainly are viruses that don't act that way. HIV would be one of them. And so we need to better understand the role of that part of the immune system. In that case, T cell populations. In addition, we need to better understand where this virus hangs out. You know, this virus clearly is new. And while it may behave like other coronaviruses, we didn't get to study those as much either. MERS and SARS were controlled. They had a higher lethality rate, and I think that's why they died out. But we didn't get a lot of experience with those coronaviruses. Yet we see a lot of different coronaviruses that can cause the common cold. But clearly, the common cold coronavirus behaves completely different than the MERS or SARS coronavirus, and I would say completely different than COVID-19. And so I think we need to find out fundamentals of where this virus hangs out, how the body's immune system takes it on, and what implications that might mean for immunity, reinfection, or disease status. But Leslie, what do you think about endpoints?
Yeah, so actually, I was thinking about this as I mentioned at the beginning of my my remarks. This is a constantly changing. The information is constantly changing. We're in a situation where the data changes daily. What we know about the virus changes, as Chuck said. And I think that this is a case where we definitely have to balance the methodology and the information. And this is exactly the kind of situation where something like adaptive designs makes a lot of sense. I'm just promoting my own research here, as Chuck knows. So this is a case where, as we learn throughout a trial, we can make changes to that trial. And certainly if there are changes in what we know about how to define an outcome, while not ideal to change your outcome during the course of a trial, we can adapt our trials to that. I think Chuck's point is really important regarding not knowing about the disease, and that impacts the trials we can do and how we can carry them out. Because if we design a trial to test a specific endpoint and then in time find that endpoint isn't truly the end, then it changes the kinds of decisions we can make and the conclusions we can draw from that trial.
Okay, thank you.
And Leslie really is a leader in this area of adaptive trials. And that's a big advance, by the way, in trial design. That's how we can get to answers faster and we get to better answers than we did before. I do think it also speaks to the fact that we need to follow these patients for quite a long time, 6 months to a year at minimal.
Right. Then the flip side of that is then that doesn't help us to expedite the process. And so having short-term and longer-term follow-ups or endpoints can be really important for decision making.
And we might be able to
Thank you.
release information in the setting of a disaster, because I think one of the key things that hasn't happened with clinical trial results is getting them into the hands of the clinicians who need to use them. And maybe we can speed up that process a little bit, but we have to be very careful so that we at least have the same rigor as we release that information.
Thank you. Another question concerns the possibility of a prophylactic medication. So for example, by analogy with HIV, we have PrEP or pre-exposure prophylaxis, a medication that someone who is not infected can take to prevent the likelihood of becoming infected if they are exposed. Is there any, are there any of the clinical trials that are exploring the possibility of a preventive medication which might be useful in advance of the potential subsequent availability of a vaccine?
Well, there are, Jim. I mean, there are lots of people who are thinking about this. I mean, that's theoretically one of the issues with hydroxychloroquine and chloroquine, is that if you can stop the virus from infecting through this mechanism of changing the pH of the vacuole that's going to bring it in, then potentially you keep the virus from infecting. These are all theoretical concerns. The other ones, with some of these anti-influenza antiviral agents have been looked at in terms of trying to prevent infections among patients who are contacts of mild cases of COVID. In very small studies, there appears to be a difference between those who receive these drugs, mainly out of Japan. And the name of the drug is favipiravir, and it's not in the United States. The data aren't strong. But the idea, again, is to try to make the environment for the literal infection from the virus to human cells be stopped. I think, though, that's a pretty tall hypothesis, because I think the challenge here is that these viruses have been evolving over eons. This particular virus has been rapidly spread, and the infectious rate of this virus has been determined to be as high as being able to infect 6 people from one person. So it's a very infectious, effective agent, and I'm not sure a single intervention will prove effective. But clearly, it's a good line of research.
Jim, I think you're on mute.
Thank you. Another question concerns who's in charge. You mentioned that there's efforts to coordinate research, particularly with the National Institute for Allergy and Infectious Diseases. But on a global scale, is there a corresponding global effort to harness and coordinate the different clinical trials that are ongoing?
I can speak from my experience. Yes, I was part of a group that you mentioned called the United States Critical Illness Injury Trials Group. We actually have counterparts in Canada, Australia, New Zealand, Japan, China, Europe. So we've been coordinating our research for a decade, frankly, ever since SARS. And so we do have a group that meets, and frankly, we had a data set and it's being uniformly collected. And we're trying to coordinate as much as we can our understanding of our clinical experience across the globe. The challenge, of course, is that treatments, as we said, there hasn't been a definitive treatment found. And so right now we're trying to take our collective experience with these other treatments and get a better clinical understanding. Leslie pointed out all the challenges of doing that, Jim. This is anecdotal experience, and while it may be large in number, it's still very individualized, and it's not done in a protocol. The World Health Organization has now launched an interventional program across those key agents. including hydroxychloroquine, including remdesivir, including these NIH protease drugs, in order to understand what the impact might be. But one of the challenges we faced is that we're doing this as volunteer professionals who've been working together. This is an area of passion. It's not an organized governmental or internationally supported effort. And frankly, we called for such an effort 10 years ago. And we have people who are engaged and will champion such an effort. It hasn't been implemented. But at some point, we're going to have to get a good understanding, because from the medical side, we want to get the right diagnosis in the right patient to provide the right treatment at the right time. And yes, we understand it should probably be at the right cost. We could do this, Jim, but frankly, the coordination and investment in such a system has lagged behind that aspiration. But we'll continue to try. I want you to know, dedicated colleagues around the globe are doing this volunteer effort, are working together, and some groups like the Wellcome Trust have funded this research. And we hope that we can continue to learn from our collective experience, but frankly provide a blueprint for the future, so we'll be better prepared for the next one. And Jim, there will be a next one.
Thank you. This is a sobering thought. I appreciate your insight on that. We have a new question that concerns questions that have been raised about the accuracy of diagnostic testing, both the potential that an infected person may have a negative test, or possibly the flip side of a false positive test. How does the potential for misclassifying someone as to whether or not they have this infection, or possibly whether they've eliminated it, how does that potential for misclassification, given the limitations of diagnostic testing, affect clinical trials? And I suspect in a way this is an extension of the previous question you answered about the situation where people tested negative and then positive again.
Yeah. So I can start with this one, and then Chuck, you can follow up from a clinical perspective if you'd like. So I believe the way the question's posed, if someone tests negative, it doesn't mean they'll necessarily be in a control group. So what it means is they actually won't be in the study. And so we'll be missing people that could be included in the potential population. So the control group would be the group of people who have COVID but aren't getting the active treatment. And so we're comparing what their outcomes are between those who do get the treatment and those who don't. So potential false negatives are people who test negative but do actually have the disease, then miss out on the opportunity to get treated. And that can be a problem from the perspective of creating future infections and sharing the disease with others, if you will. But from the perspective of trials, it makes the pool smaller.
I mean, first of all, I like that answer from Leslie. I guess the only clinical perspective I'd bring forward is that there's not only uncertainty in terms of how the virus would be detected and where it hides out and the timing of that test, which could be critical, of course, but also the technology of the test used. I mean, the classic is to use, RT-PCR. This is, reverse transcriptase polymerase chain reactions. This is a way to directly look at the viral RNA and then detect it directly in the sample, whether it's blood or sputum or from the nose or anything. There are a number of other tests being proposed, and some of them have been approved by the FDA, that use just pieces of that RNA, and those are called molecular tests. They have the advantage of being rather rapid, but they won't have the same sensitivity. They won't be able to detect every positive test, and that could influence then, the patients who get enrolled in a trial as positives. But I really liked Leslie's point that, just because if they're test negative, then they might not make it in the trial. So imagine if we're taking the results of a trial that used a technology that was only specific at identifying infections using that particular brand of test and try to extrapolate out to a broader group, we may not have the same answer implication.
And I'll just add that one of the things I love about chairing an epidemiology and biostatistics department is I get to work with really, really intelligent people. And we have regular check-ins with our department. And actually this morning on our Monday morning check-in, we did have this conversation about false positives and false negatives. And one of the concerns, as Chuck raised, is that there's no gold standard. There's no way to determine that test is good or bad relative to something that's a sure thing every time. And so that makes it a little more challenging to determine who is a false negative and a false positive. At what point, if someone's tested and retested, may they show positive, and what factors influence that? How much virus they're shedding or their viral load, how much does that influence the test? So there's still a lot of information we don't know that could inform who a new treatment might work on.
Thank you. Another question concerns desire to have information about what might be safe and effective in treating pregnant women who have COVID-19 infection, and that raises a number of issues. First, about how does the inclusion potentially of pregnant women in a study affect the ability to do studies quickly, but also there's clearly unique safety concerns about drugs and particularly drug testing in pregnancy. Speaking from my own perspective as a pediatrician, similar concerns often arise about how do we know that drugs that are tested in adults are going to work in children, and yet it's more complicated to do clinical trials in children. So can you speak to these special circumstances of testing drugs in the situation of pregnancy or for children?
I'll be happy to start then. It's a challenge, Jim. We know that pregnant women are a special category because they are susceptible. And they're susceptible, one, because there are changes that occur with pregnancy, including how drugs are metabolized. There are other hormones, obviously, at play. And the body's defense system is different during pregnancy. We know this can particularly affect viral infections. And of course, the classic is measles. And measles in pregnancy can be a very disastrous combination. I think that, so there's that one, the inherent changes that occur to women when they become pregnant. The second issue, of course, is there's a fetus. And so now you have, fetus is developing. It's very susceptible to changes. So drugs can influence that development. The infections can behave differently in fetuses. There are lots of questions right now about COVID and its transmission to fetuses. There's also questions about COVID as, and how it's transmitted in breast milk. And these are similar to the questions that occurred, during Zika and other viruses like SARS and MERS before that. And so these really do have to be considered as special cases. I think from the clinical trial standpoint, the reason that women and children are in different categories is because, number one, the consequences of a bad outcome or reaction in a pregnant woman or a child can be devastating. And at least from the medical ethical side, the dictum of do no harm really is a driving force. And so you have to weigh the possible consequences with any particular benefit. And I think it becomes particularly acute in the case of pregnant women and children. The second piece is we don't have a lot of the data on all those factors that are different between women, in some cases in children and adults. And I would say that's particularly true of this virus. There aren't very many children that are being affected. And so the clinical experience with this virus is even less informed than it is with the adult side. And the challenge with that, of course, is that we don't know a lot about it in the adult side right now. So people are typically quite conservative. But I hope that, and we know there are people, though, who are going to become desperately ill, who are pregnant, and those who are children. These experimental treatments are going to be tried, I suspect, out of clinical desperation and out of an individual physician trying to balance that cost versus benefit ratio. But Jim, it's a tough challenge. It's one we have to recognize. The NIH certainly has rules about it. And I don't know if Leslie wants to comment further on how it's approached systematically in clinical trials.
Yeah, I think more so just about the information we have. And I know that there are several cohorts out there that are collecting data specifically on women who are pregnant and their experiences with COVID to help inform future trials, should we need them and should they occur. So that, as Chuck mentioned, we just don't know. We don't know how this might express itself differently among women who are expecting, what the implications are for the fetus and the yet unborn child, and then for the transmission in breast milk. And so there are studies that are looking, not clinical trials, but other types of studies that are collecting data and information about the disease in pregnant women to help us inform future trials.
Thank you. So we have a few more questions, but I'm afraid we're getting very close to the end of our hour. One last question, if maybe one of you has a quick answer. I think in many respects we've already addressed this, but the question is, how are people selected to get into a clinical trial? And are certain regions or certain hospitals specifically targeted?
I'll answer really quickly from the critical care emergency side. They have to have research infrastructure in place to be able to do these studies. Clinical care in COVID-19 is complicated. You have to have personal protection equipment. You have to have protections for the staff, patients. The families are typically excluded from the hospital except at very specific times. So it's a very complex environment to begin with. You've got to have dedicated research infrastructure with people who are trained, knowledgeable, and experienced in conducting research in order to do research. So that is kind of an attention, at least in the hospital critical care arena.
And then just one additional, you have to have a sufficiently sized patient population that can to be included in the trials often. So if you're not seeing a lot of patients, then it's possible you might, if that area is not seeing a lot of patients, it's possible they might be included in the trial, but more likely they would, study sites are selected based on their patient flow, on the number of patients they see. And as I mentioned, actually with something like COVID, where the wave of the epidemic is shifting from place to place, that could impact how trials are happening in a very interesting fashion that actually could lead to some really interesting research.
Well, we're almost out of time, but before we wrap up, I'd just like to invite each of you, if there's any summary comments or additional points that you'd like to make before we end the session. Perhaps Leslie, I'll let you go first, and then Chuck.
Thank you. Just to say that there's a lot of very excited, energetic, intelligent people who are thinking about these problems, and we're really fortunate to have a strong community of clinicians, of epidemiologists, statisticians, of healthcare workers who are ready and at the forefront of this, trying to ensure that we all have the best outcomes we can. And it's done through a mutual desire to improve the health of our communities and of our public. And so, it's always been my honor and privilege to be a part of that community.
And Dr. Cairns.
Jim, it's a pleasure and honor to be part of this panel. I just want to assure everyone who's watching or everyone who's participated that our clinicians, our scientists, our staff are all dedicated to taking on the challenges of COVID-19. They're experienced, they're dedicated, they are absolutely looking for the right answer, not only for the right patient, but for general, to keep our knowledge base sound as we move forward together as a region, a nation, and a world to combat this virus. And as I said, challenges that will come forward. We've made remarkable gains in clinical trials, management, analysis. The tools available to us in biotechnology are remarkable and unheard of even 10 years ago. And I do think that we have an ability to safely and effectively evaluate these treatments and come up with optimal, if not maximal, solutions on how to best treat people. So thank you so much, Jim, for the honor to be in this panel today.
Yeah, thanks, Jim, for being our moderator. We appreciate your guidance and your input.
It's a pleasure to be with Leslie. I learned something at every one of these sessions. Thank you.
Well, I learned a tremendous amount today. I'm sure that the audience learned a tremendous amount as well. Let me thank all of you who are tuned in for connecting with us this afternoon. Let me thank the two of you for excellent presentations and very insightful and informative observations. And if you're interested in continuing to listen to and watch our webinars, check www.drexel.edu/Dornsife for the list of upcoming sessions. Have a great afternoon, and thank you again.
Thank you.
Stay safe.